So this is the muscles and muscle tissue chapter. We'll talk about the physiology of muscle contraction along with the anatomy of the muscles and the muscle tissue. Now, a skeletal muscle cell is also called a skeletal muscle fiber. We learned in lab, we saw them, they were these long straight fibers. And within those fibers, we have these things called myofilaments, and we're going to see actin and myosin are going to be two proteins, these two filaments that are in there that are going to interact with each other to contract the muscle. Some other terminology to be familiar with the muscles. When we talk about the muscle cell membrane, it's called a sarcolemma. When we talk about the cytoplasm in a muscle cell, it's called sarcoplasm. Now, whenever you see the prefixes myo, mes, or sarco, they always or almost always refer to muscle. Sarco actually means tissue, but it's almost always used to describe muscle. Aspects of a skeletal muscle tissue, well, we learned this in lab too. They are striated, it is voluntary, right? It's under conscious control, it has multiple nuclei, and it's responsible for movement and locomotion, right? So this is what provides movement of the skeletal structure, in your body, through movement through the joints, range of motion. Cardiac muscle is only found in the heart, and it's not including the blood vessels that are in and come out of the heart, but it's just the actual heart tissue, and 95% of the heart is actually made up of cardiac muscle. It's also striated, however, it is involuntary. It only has one nucleus, so it's uninucleate, and the cells are separated by these discs called intercalated discs. Smooth muscle, not striated, is also involuntary like cardiac muscle, also uninucleate like cardiac muscle, and it's found in a number of visceral organs. The key aspects that I usually like to focus on, blood vessels have them, the digestive system, but the smooth muscle is found in a number of other visceral organs as well. Again, some quick terminology and differentiation between terms. When we talk about excitability, we're talking about the ability of something to respond to a stimulus, so its responsiveness. Sometimes it's called its irritability. Contractility is the ability of the muscles to shorten, and the extensibility is the ability to extend or be stretched. Elasticity is the ability to recoil or bounce back from that stretch. Now, when we look on this slide, we know that locomotion is the function of skeletal muscles, as I said before, cardiac muscles to pump blood, and then the peristalsis, which is that rhythmic contraction of muscles that we've talked about in other chapters. That's what's going to propel things like blood in the blood vessels or food within a digestive system. When we talk about other functions of muscles like maintaining posture and stabilization, we're talking about skeletal muscles. And we'll talk about the generation of heat from the activity of these muscles as well, and the percentages later on. Coverings of a muscle, well, epi means upon, peri means around, and endo means within. So epimysium, this is actually the covering around the entire muscle. Then you have perimysium. This is the covering around groups of muscle cells or muscle fibers called fascicles. And then the endomysium, endo means within, this is the surrounding of each individual muscle cell, aka each individual muscle fiber. And this picture gives you a nice visualization of the muscle fiber, fascicle, and muscle with their coverings. How do we attach muscle to bone? Well, it directly attaches from the epimysium, fusing with the periosteum of the bone. It also indirectly attaches with tendons and sometimes with these membranous sheaths called aponeuroses. Just another good visualization of looking at the muscle, pulling out a fascicle, which is a group of fibers, and then pulling out a fiber that actually has those little myofibrils in them. So what's a myofibril? It's a rod-like contractile element, and we're going to be talking about these when we talk about muscle contraction. Let's go ahead and look at a picture of this. So you can see the skeletal muscle. This is one skeletal muscle cell / fiber, right? Same thing. And you look inside and you see myofibrils, these long cylinders inside. And then you see how they're sectioned. Well, each of those sections is called a sarcomere. So the sarcomere goes from what's called Z disc to Z disc, and we're going to have these filaments in there, these proteins, these myofilaments called myosin and actin, and we'll be talking a lot about those. Now, if you look on this picture, you can see the Z disc to Z disc. Each one of those is a sarcomere, so you have a bunch of sarcomeres strung together, making up these myofibrils. And then when we look at the filaments, you can actually see the yellow guys down at the bottom there. Those are actually, we don't talk too much about them, but those are anchoring filaments called titin. Those are elastic filaments. Then you have the blue, which is the actin filaments, and then the thicker ones, which is the red. Those are the myosin filaments. So myosin is the thicker filament that has a rod-like tail, and it has two heads. You can see a picture here of myosin with the heads and tails, and we'll see how they interact later with actin. Actin is the thinner filament, and it's going to also interact with the other filament, myosin there. It has some structures on it called Tropomyosin and Troponin. We'll look at that on the picture. If you look here, you see the blue filament, right? That is the thin filament called actin. Then you can see these orange bands. Those orange bands are called tropomyosin, and they're covering up little active binding sites underneath there. And then you got this yellow thing called the troponin complex. We're going to see how that all works together in just a little bit. This is just another really good visualization of the actin and the myosin and their structural components. On this, you can actually even see, if you look down towards the bottom right there with the actin subunits, you can see the little active binding sites for myosin's heads to attach to. And so we'll see how that works in just a little bit. So in the muscle, it has endoplasmic reticulum, specifically smooth endoplasmic reticulum that is called sarcoplasmic reticulum. Muscle likes to have its own names, right? And these are going to store calcium, and they're going to release calcium from these ends called terminal cisternae. We'll see that shortly. We also have these things called T-tubules, and they're going to carry the impulse deep into the muscle fiber so that all of the myofibrils get stimulated, because when a muscle cell, which is a muscle fiber, contracts, it's an all or none thing. So all of it contracts, including all of the little myofibrils in there and the filaments interacting with each other. Now, this is where I showed you, or said in the last slide, that the calcium releases from terminal cisternae of the sarcoplasmic reticulum. And what's the stimulus for that? It's the impulse coming down the T-tubules. That's what's causing those voltage gates for calcium to open. We're going to see all this in detail, but that's what's happening. So let's look at some of the structure here. See those yellow guys? Those are the T-tubules. They're going to carry the impulse down throughout all of the myofibrils. Do you see the purple stuff? That is the sarcoplasmic reticulum, and at the ends of it are the terminal cisternae, where it's going to release calcium. Why does it release calcium? Well, it's the stimulus coming down that T-tubule, that voltage, that's going to open those calcium gates and allow the release of calcium. Now, when we talk about this sliding filament model, we're talking about the action and the interaction between myosin and actin. And what's going to happen is the little myosin heads are going to attach to those binding sites of actin, and they're going to ratchet inward, and we're going to pull the myosin, those blue filaments on the picture, we're going to pull those towards the midline. And so what it's going to do is it's going to shorten the sarcomere, and that's going to happen throughout all the sarcomeres, which is going to shorten that muscle fiber. So you see here, you've got a little bit of overlap between myosin and actin, but then as those little heads are going to act to ratchet the actin filaments towards the midline, you see how it pulls those filaments in, and it shortens that sarcomere. So this won't make a ton of sense yet, but it will once we go through the whole process. In order to contract a skeletal muscle, we have to stimulate it with a nerve. There's an electrical current, which we're going to call action potential in the muscle. It's going to travel down the muscle cell membrane called the sarcolemma. It's going to be taken deep into the cell by the T-tubules that we mentioned before, and then there's going to be this rise in calcium as we release it from the terminal cisternae, and that's going to have an intimate role in this contraction process. So this slide talks about the neuromuscular junction. Now, the neuromuscular junction is made up of the axon terminal and the motor end plate of the muscle cell membrane, which is the sarcolemma. So there's a portion of the muscle cell membrane called the motor end plate that has the receptors on it. It has these receptors called acetylcholine receptors, because the neurotransmitter acetylcholine is going to be what's released from the axon terminal. It's going to cross this little gap, this little cleft, and it's going to attach to those receptors. So basically the axon terminal plus the motor end plate together makes up the neuromuscular junction. So let's go through the process on this slide. You can see the impulse coming down the axon into those little axon terminals. Now you zoom in now, you see one of the axon terminals, and you see the motor end plate of the muscle, which is part of this cell wall called the sarcolemma, remember, in the muscle. And so we've got that axon terminal. As the impulse hits that axon terminal, you're going to see calcium channels in that axon terminal open up. And those are voltage gated channels, right? So that when the voltage comes down and hits those gates, they open, calcium floods into the axon terminal, and that triggers a release of those little green things. That's acetylcholine. So you see those little vesicles containing acetylcholine, they will bind to the receptor, they will bind to the cell wall, release the acetylcholine into the synaptic cleft, which will cross the synaptic cleft, which is the gap between the two, and then bind to the receptors on the muscle cell, which is on that motor end plate. Those little purple guys are the receptors, okay? So you see that process happening here. It gives you all those processes. Now what happens when we bind that chemical, that neurotransmitter called acetylcholine, to the receptor on the motor end plate? Well, when we do that, the gate opens, right? This is a chemically gated channel. So the chemical is the key that opens this gate. And when it opens the gate, you've got sodium flooding in and potassium going out because that's the direction of their electrochemical gradients, okay? So this talks about what I was saying on the other slide, how when the impulse hits the axon terminal, it releases or opens the calcium gate, calcium floods into the axon terminal. That's going to cause the release of acetylcholine. When acetylcholine gets released, it crosses the synaptic cleft and binds to the receptors, the acetylcholine receptors that are on the motor end plate of the sarcolemma. Now you can also go back to that picture and you can look and see that when we're done with acetylcholine, we actually use acetylcholinesterase, which is an enzyme that breaks down acetylcholine. And so we want to make sure when we're done contracting, we stop the contraction. So this enzyme breaks down acetylcholine to cease the contraction. Botulinum toxin, this is something that blocks the release of acetylcholine from synaptic vesicles. You can get it from improperly canned foods. That's one area you can see this toxin. But it's also used medicinally as Botox, which is used for cross eyes, uncontrolled blinking, if there's spasms of the vocal cords. Also for wrinkles it is used, as well as sometimes for muscle spasms with chronic back pain, most often in the lumbar region. Curare is a plant poison that actually causes paralysis by blocking acetylcholine receptors, and so it inhibits those sodium ion channels. And derivatives of curare are actually used during surgery to relax skeletal muscles. There's something called anticholinesterase. What this does is slows down the actions of the enzyme acetylcholinesterase. So basically what it's doing is it's slowing down the removal of acetylcholine. So this will oftentimes be used to treat myasthenia gravis. It can be an antidote for curare poisoning. Can also terminate the effects of curare if it's used for surgery. Now, a local depolarization is when we use that acetylcholine that crosses the synaptic cleft, binds to the receptor, and opens the sodium-potassium channel. We're going to generate a local depolarization. Depolarization means we're going to move to a less negative state inside the membrane. So the resting membrane potential for a muscle is somewhere in around negative 90, and we're going to move to a less negative state by allowing sodium and potassium to move in and out. Now, sodium was mostly out of the cell, so it will move into the cell because things like to move from high to low. Potassium was mostly inside the cell, so it will move out of the cell because it moves from high to low, high in to low out. Now, they're both positive, so you say, well, how does this make the membrane inside less negative? Well, more sodium is actually going to come in than potassium going out because we also move electrically to the opposite charge, which means both sodium and potassium are going to want to move inside electrically. So that pull on potassium to stay inside electrically is going to cause the influx of sodium to come in more than the potassium going out. So the electrochemical gradient for sodium is stronger than the electrochemical gradient for potassium. So in order to get an action potential, we have to reach a threshold. So basically we have to get to a certain point of less negativity as we're depolarizing. And so as we depolarize the membrane, adjacent patches of that membrane become depolarized, and we're opening sodium gates. These are voltage gates or voltage channels that open. And so more sodium is going to flood in. When we hit this threshold, then we generate an action potential where all the sodium gates open, lots of sodium floods in, and we move all the way to the way less negative, past zero, into the positive numbers. And so that is the generation of our action potential happens at that threshold. Once we completely depolarize, right, we become less negative and then eventually more positive, we're going to repolarize the membrane. So the depolarization with sodium flooding into the cell, then we're going to close the sodium gates, we're going to open potassium gates, right, in that somewhere around that positive 30-ish number there, and then we're going to And again, you have to know that number for this, and it might be slightly different for a muscle than a nerve. But essentially what's going to happen is somewhere in those positive numbers, we're going to hit a certain voltage where we close those sodium gates, we open the potassium gates, and potassium is going to move out of the cell. Well, this is going to repolarize the membrane. Repolarization means that we're actually going back to the resting membrane potential. From this positive state, we're becoming more and more negative until we get back to our resting membrane potential. Now, when we get to the resting membrane potential, the charge is restored. So what do we need to do now? Well, we need to use a sodium-potassium pump to pump the ions back to where they were. Because what happened? Sodium flooded in when we opened gates, potassium flooded out. So now we need to pump them against their concentration gradient. So the sodium-potassium pump will pump sodium back out of the cell, back to where it came from, outside, and it will pump the potassium back into the cell. This picture shows the local depolarization and how we're going to open in adjacent patches these voltage-gated sodium channels, moving the membrane, moving this charge down the membrane. And again, as sodium floods in and we hit a certain threshold, we're going to depolarize or create this action potential that'll depolarize the entire muscle cell. And then we'll get that contraction of the muscle. Then we have to repolarize the muscle cell, like I said before, by closing those sodium gates and opening those potassium gates. Now, I mentioned this earlier in the chapter. We want to carry that action potential all the way down the cell membrane, but we also want it getting deep into the cell so we hit all the myofibrils. And so the T-tubes are going to take that voltage down deep into the cell, right, throughout those myofibrils. And what it does, and you're going to see this on a picture, is it's going to open calcium gates, and that calcium is going to release, and we're going to use that to bind to troponin, which is going to move tropomyosin out of the way. I'm going to go through this detailed process here, and that's going to allow the opening of those binding sites on actin. Now we're going to go through this on the pictures, but we're going to see that the myosin heads are going to bind to the actin active binding sites. That's going to cause what's called a cross-bridge formation, and then it's going to ratchet and it's going to pull those myosin filaments towards the center line. Now, we're going to use ATP. When we break ATP, that's what actually cocks the myosin head. And then we'll talk about this a little bit more detail in the pictures. So if you look here, now you see that the impulse is being taken down the T-tubules, and it's going to pass through the sarcoplasmic reticulum that contains the calcium. When it does that, it opens calcium gates. I'll show you on a different picture, but it's going to open calcium gates. And that's going to release the calcium out of the terminal cisternae of that sarcoplasmic reticulum. And if you look down here, you can see down here at let's see, what is it, number 3 and 4, you can see that the calcium is going to bind to that little yellow guy, that's troponin. When it does so, it changes the shape of troponin that actually pulls the orange band, which is tropomyosin, out of the way, because what tropomyosin is doing is it's covering those binding sites. When that happens, we're able to bind that myosin head like you can see in number 4. When we're done, we'll ship the calcium back into the sarcoplasmic reticulum. And the removal or the separation of myosin from actin is actually the binding of another ATP causes them to separate. And then we're going to break that ATP to get the energy to recock the head. Again, this is something I'll be covering again on a different picture. This shows a nice close-up of the voltage gates for calcium. As the T-tubules carry the voltage, they open those gates. The calcium gets released out of the terminal cisternae. And that calcium is the calcium we're going to use to bind to troponin to change its shape. So you can see the calcium, right? It binds to the yellow guy, troponin, and you can picture that protein actually changing shape as calcium binds to it, pulling the tropomyosin, the orange band, out of the way. So let's go through all these steps. The cross-bridge formation, the Powerstroke, the detachment of the cross bridge, and then the recocking of the myosin head, and we'll go through that on each picture. So we know where we're getting the calcium from, right? It was the T-tubule sending the voltage down, opening the voltage gates for calcium, coming out of the sarcoplasmic reticulum. We had this calcium, the little orange balls. And so those calciums are going to bind to the yellow guy, as I said before. That's troponin. And then it moves tropomyosin out of the way. So you can see on this picture we have what's called a cross-bridge formation. You see the head of myosin binding to the active binding sites, those little half circles. It's binding to those sites on actin, so they're connecting. Okay. Now the Powerstroke is when myosin ratchets inward. So those little heads will ratchet inward and will pull actin towards the midline. When ATP, another ATP, attaches to the head of myosin, it causes the head of myosin to disassociate or unbind from actin. And then lastly, we're going to have the cocking of the myosin head. So as we hydrolyze ATP, so hydrolysis of ATP is going to release energy to recock the myosin head. If the binding sites are still open, this process will repeat, and the myosin head will rebind to actin, ratchet forward again, or ratchet inward again, and then detach when another ATP attaches to the head of myosin. So this cycle will repeat as long as calcium is available and bound to troponin, keeping the tropomyosin out of the way. We will have this process happen. So isometric contraction versus isotonic. Isometric contraction, the muscle tension increases but doesn't exceed the load, so the muscle does not shorten during this type of contraction. Isotonic contraction, the muscle does shorten because the tension exceeds the load, so you're getting that shortening during contraction. Those are the differences. So like an example of an isometric contraction would be like holding a push-up or a plank position. An example of isotonic would be doing a biceps curl or a triceps extension or something like that, moving the muscle through the range where it's actually shortening. That's the isotonic. Now, a motor unit, what is this? A motor unit is a motor neuron and all of the muscle fibers that it supplies. We'll look at a picture on the next slide. But there can be, from one motor neuron, there can be up to 4 muscle fibers, which are muscle cells, or there can be several hundred. Now, when we look at this, muscles that control fine movements like the fingers and eyes are going to have smaller motor units, and larger, stronger, bigger weight-bearing muscles like the thighs and hips are going to have larger motor units. So on this slide, we can see the motor units. We've got motor unit 1 and 2 as examples that we're giving on this. And the purple, you can see it attaches to, that one motor unit attaches to 2 fibers, and motor unit 2 attaches to 3 fibers. Obviously, they're keeping this very simplistic. But the thing to understand is that there are separate motor units. Now, a muscle fiber, we said, contracts all or none, right? The entire, if we stimulate a muscle fiber, the whole thing contracts. Well, it's the same for a motor unit. If we stimulate that motor unit, all of the muscle fibers attached to it will contract. So for motor unit 1, both of those fibers. For motor unit 2, those 3 fibers will contract completely if that motor unit is stimulated. All right, so a couple of key aspects about these motor units. The fibers from that motor unit, they're not all packed together. They're spread throughout the muscle, so you get a smooth, homogeneous contraction. Again, if let's say all the muscle fibers are packed on one side, when you contract, it would yank the muscle to the one side, right? And so the fibers are spread throughout, and so it causes a weak contraction of the entire muscle when you stimulate that motor unit. How do we make it stronger? Well, we're going to recruit more motor units, and we'll talk about that in just a little bit when we talk about recruitment. Now, motor units in a muscle usually contract asynchronously to help prevent fatigue. So what happens is we'll contract one motor unit, then another, then another, then another. And so we're able to continue muscle contraction without keeping that motor unit activated so that it delays fatigue. So some stages of muscle contraction we have, or phases we could say, of a muscle twitch. Now, what is a muscle twitch first? It's a single contraction, a single response of a muscle to a very brief stimulus. Now, the events in contraction, we have the excitation-contraction coupling, which we cover the detail of with the nerve sending an impulse down into the axon terminal, the acetylcholine crossing the cleft, generating a local potential at the motor end plate. The period of contraction is actually the cross-bridge formation. And then we have a period of relaxation where we're removing the calcium from the troponin, as we saw before, and putting it back in the sarcoplasmic reticulum. Now, graded muscle responses. This is the variations in the degree of muscle contraction, and in order to have proper control of skeletal muscles, we need to be able to grade it, right? In other words, if I'm going to lift an empty cup compared to, let's say, a jug filled with water, right, or lift a pen compared to, say, a 20-pound weight, I have to change the amount of force that I'm generating from that muscle, right? Now, how do we do that? How do we change the muscle response? Well, we can change the frequency of the stimulation, or we can change the actual strength of the stimulus, and we're going to look at these a little more in depth. Now, what you see on this one is that we have a single stimulus, which we said was a muscle twitch. But if we send the stimulus more frequently, so before the muscle has time to completely relax, like in that very first one, okay, we're going to summate. In other words, we're going to send a stimulus so increase frequency of it so that before the first stimulus has completely gone away, right? The contraction has stopped. We send another stimulus, and you can see how it rises, and the peak is a little bit higher on that second one. And then, of course, we're going to talk about on the next slide as we increase the frequency even more. On this slide, we see incomplete tetanus, where we increase the frequency of the stimulus even more. You can see how the stimulus is getting closer and closer together, right? The time between is shorter and shorter. And then, of course, in the fourth, which is called complete tetanus, the stimulus is just firing so fast that there's no relaxation whatsoever, so we get maximal contraction of the muscle. Now, I mentioned this term threshold, and the threshold stimulus is the stimulus strength at which we see the first observable muscle contraction. We said if we don't reach stimulus, if we don't reach threshold, we're actually not going to, create an action potential, which means we're not going to contract the muscle, right? We have to generate the action potential. When we do that, that's where we're going to get that complete depolarization of the entire skeletal muscle cell, which is going to cause the skeletal muscle to contract. Now, the force of the contraction is controlled specifically by motor unit summation. What does that mean? That means we add more motor units as we want to increase the force of contraction. This is called recruitment. What we're doing is we're recruiting more motor units, which means we're recruiting more muscle fibers. And the more muscle fibers we contract, the more force that contraction has. Now, we said in addition to increasing frequency, we can increase the stimulus. So as you look on this, that first top picture there, right? So 1 going all the way through 10, you can see the stimulus increases and increases and increases. It gets to a point of maximal stimulus. So when you look at number 7, it hits the maximal stimulus. So even if we go above that, you've already hit the maximum number of motor units that you can activate and muscle fibers that you can contract. And so if you look at the middle picture, you see the red is the number of muscle fibers being activated, or at least motor units being activated. And then, that gives you an idea that the stronger that stimulus that comes through, the more fibers are going to be activated, and then you're going to get better and stronger force of contraction. So when we talk about muscle tone, this is a slightly contracted state of muscles. So when we're sitting here, right, if we're sitting upright, we have postural muscles that are maintaining muscle tone. And it's the spinal reflexes that account for this muscle tone. So they activate one motor unit, then another motor unit. And we talked about how to prevent fatigue, we're going to asynchronously contract motor units. Well, not only are postural muscles a good example of very slow fatigable muscles, but we're also going to use this principle of activating different motor units so that, in 5 minutes we don't just get fatigued and fall over like a bag of jelly, right? Now, we mentioned isotonic and isometric contractions before, and we said that isotonic contractions, that we are changing the length of the muscle and we're actually moving a load. So I mentioned biceps curl, I mentioned triceps extension, even picking a book up off a table is another example. There's 2 phases, though, to the isotonic contraction. There's the concentric and the eccentric. The eccentric is sometimes known as the negative. The concentric contraction is when the muscle is shortening, and the eccentric is when it's lengthening. Remember, we don't just contract the muscle and then poof, it just blows out, right? We contract it on the way up, making it shorter, and we contract it as it's lengthening, right? So there's contraction through both phases, through the shortening and the lengthening back to its resting state. The shortening is the concentric, the lengthening is the eccentric. And we already mentioned isometric contractions where we're not changing the muscle length and the tension generated. So examples like a push-up or holding a push-up, not moving through a push-up, that would actually be isotonic, but holding a push-up or plank position, holding an object, with an outstretched arm, things of this nature. Now we know that ATP is our source of energy, and that's what we're going to use for contraction. It's actually what we saw cause the cocking of the myosin head, right? Or the hydrolysis of that ATP. And so we have different sources of ATP. We have stored ATP, that gives you about 4 to 6 seconds of energy. But then we're going to have 3 other systems we'll talk about. So we have in the muscles some stored creatine phosphate. And every 1 creatine phosphate has the ability to produce 1 ATP. So this is another, so we have the stored ATP, we have the creatine phosphate that can be converted, 1 creatine phosphate to, basically 1 ATP per creatine phosphate. Now, an anaerobic pathway means we don't use oxygen, and so we can generate energy from using oxygen or not using oxygen. And at 70% of your maximal contractility or contractile activity, that is going to be where we use the anaerobic pathway. You're going to see muscles bulge and compress the blood vessels, impairing oxygen delivery, And this is where we're going to get pyruvic acid converted to lactic acid. This is something we cover in depth in Chapter 24 in Anatomy 202. But for now, this is enough. Some examples: sprints, soccer, tennis, weightlifting are examples that would use anaerobic pathway. Now we do have something called the Cori cycle that's going to ship the lactic acid from the muscles to the liver. We're going to convert the lactic acid back into pyruvic acid, and then that pyruvic acid back into glucose. So what is this anaerobic pathway I'm talking about? It's called glycolysis. Glycolysis is the breakdown of glucose. There are 10 steps. We break down glucose to 2 pyruvic acids. It doesn't say 2 there, but I put a little 2 in red. And then if we don't have oxygen available, then we convert that to lactic acid, which it will wind up going through that Cori cycle I mentioned on the previous slide. What's important? Well, it's important to know that this is anaerobic. It's important to know that it happens in the cytosol, which is what? It's the fluid portion inside the cell. The rest of these energy systems that we're going to see, which we're going to group it together as aerobic respiration for now, but there's 3 other processes or steps that will happen in the mitochondria. Okay, so we have 2 ATP that we're gaining from this process. Now, it's a net gain. It costs us 2 to do this process. It's a 10-step process, but we get 4. And so really, we technically get 2 is our net, and that's really what I want you to focus on, the number 2. So we get 2 ATP from every 1 glucose through the process of glycolysis. Now, the aerobic pathway, we get about 95% of our ATP. When you're doing light and moderate exercise, you're going to be using this pathway. And so let's look at what we're talking about with this aerobic or oxygen-using pathway. Now we can see here, now at the top they're still showing glycolysis because you need pyruvic acid in order to do the next 3 steps. Where there's an intermediate step, there's a Krebs cycle and oxidative phosphorylation, which again, that's not something I'm covering in 201, but in Anatomy 202 I will be covering that. But on this picture, we are picking up with aerobic respiration where you see pyruvic acid. What we're going to do is a number of things that will happen. I don't want to get into all the details of that in this chapter. I want you to focus on through this aerobic process. We look at it, we see that we have oxygen available, okay? And it's happening, we see in the mitochondria. You see the picture of the mitochondria there. Now, the degeneration of 32 ATP is actually from the very beginning to the end. So if you look what I wrote on the side, the total is 32 ATP from 1 glucose. That includes glycolysis, which was anaerobic. The other 30 come from the aerobic respiration. Okay, so 30 from aerobic respiration, 2 from glycolysis, 32 total from 1 glucose. Make sure you memorize those numbers. Now, muscle fatigue is the inability of the muscle to contract, right? And this is almost never, it's rarely due to a lack of ATP. If there's certain diseased processes or conditions, it could cause, it could cause contractures, and it could be due to a lack of ATP. But fatigue itself is generally ionic imbalances and damage to sarcoplasmic reticulum. And calcium is an ion as well. So the regulation and release of calcium is impaired due to the damage to the sarcoplasmic reticulum. You also have imbalances, some losses of potassium that can disturb the ionic imbalance. And remember, we need sodium in, potassium out to depolarize and then repolarize the membrane. And so when we have these imbalances in electrolytes, that is going to cause that muscle fatigue. So now we've got to pay back some oxygen here. The oxygen deficit is essentially the oxygen we need after exercise to replenish our oxygen reserves, to restore our glycogen storage. So that process that I talked about in the Cori cycle, converting the pyruvic acid back to lactic acid, so that's kind of linked down there with number 4. But that process of doing that and then converting the pyruvic acid back into glucose and packaging it into a stored form of glucose called glycogen, which is found in the muscle cells, liver and muscles. And so we're going to need oxygen to do that. So replenishing our glycogen stores, our oxygen reserves, as well as replenishing our stored ATP and our creatine phosphate. Like all of these are things that need to be replenished after exercise. Now, a principle about muscle fatigue is that intense exercise that produces rapid muscle fatigue will also exhibit rapid recovery, whereas low-intensity exercise that takes much longer or slower developing fatigue, that may require several hours to actually completely recover from. Now, it may not seem like an efficient system that 40% of the energy released in the muscle activity is useful for work. That's actually a very efficient machinery. 60% is given off as heat. So memorize those numbers as well. Okay, this slide is talking about the things that affect the force of muscle contraction. Now, we know that if we recruit more muscle fibers, so the more muscle fibers we have, the stronger the contraction. If you jump, that was number 1. If we jump down to number 3, talk about increasing frequency allows for more effective transfer of tension to non-contractile components. So we're actually going to recruit more muscle fibers by increasing frequency of stimulation. We said that if we increase the frequency or the force of the stimulus, both of those would recruit more muscle fibers. Number 2 talks about the relative size of the fiber. So as a muscle hypertrophies, that means the muscle cell gets larger. The larger the muscle cell, the stronger it is. And then there's a length-tension relationship. So muscles contract most strongly when they're between 80 and 120% of their resting length. And this just shows the 4 increasing number of muscle fibers, the larger muscle fibers, the higher frequency of stimulation, and then being over 100% of its resting length. These are things that will all increase contractile force in the muscle. This slide shows the 3 primary fiber types: slow oxidative, fast oxidative, and fast glycolytic. Now, there are more subcategories, but this is what we're going to cover in this particular chapter in class. And everyone's muscles contain a mixture of the 3, and that is dependent on your genetics. So some people have more slow, we call them slow twitch fibers, and some people have more fast twitch fibers depending on their genetics. We can modify these with training, but understand that a fast twitch fiber will never be a slow twitch fiber, and a slow will never be a fast. So we can't cross categories like that. Here we look at some differences between the fibers, and I have some things underlined that I want you to focus on. Slow oxidative fibers are the slowest with speed of contraction. Both fast oxidative and fast glycolytic are fast. As far as fatigue, slow is the slowest to fatigue or the most fatigue resistant, whereas the fast oxidative are somewhere in the middle, and then the fast glycolytic are the fastest to fatigue. The types of fibers or types of activities we use slow twitch fibers for are going to be things like running a marathon, postural muscles. Fast oxidative fibers are going to be things like sprinting and walking, and fast glycolytic would be explosive movements like hitting a baseball or doing a weightlifting snatch or jerk or something like that. And so those are 3 different fiber types and some differences between them. Now, we can convert fast glycolytic fibers into fast oxidative fibers with using different types of endurance exercise. We can convert the fast oxidative fibers into fast glycolytic fibers when we're doing resistance training, right? So things like weights and dumbbells, barbells, machines, cables, kettlebells, things like that. We can those are even body weight movements, push-ups, pull-ups. These are all types of resistance exercise training. Now, it is important to understand the overload principle. Forcing a muscle to work hard promotes it to increase strength and endurance. So what's going to happen is it's going to plateau if we continue to do the same thing and we don't continually overload it. So the muscles can adapt to those increased demands, and we want to overload them by either increasing the number of sets or the number of reps or the weight, or changing something to continually, if you want to think about it this way, continually shock the muscle and make it continually adapt. So forcing the muscle to continue to adapt so that it doesn't plateau. Now, this term disuse atrophy, this is a degeneration of the muscle when you have something like bed rest or some kind of type of nerve damage, so you have loss of neural stimulation. And the muscle strength in this case, right, so it's not just you don't work out that day, it's if you are bedridden or you do have neural damage, the muscle strength can decrease at about 5% per day, and it's called disuse atrophy. Muscular dystrophy is a disorder. It's actually a group of inherited muscle-destroying diseases. And the muscles get larger, but it's due to fat and connective tissue deposits. The actual muscle fibers themselves will atrophy. The most common type of muscular dystrophy is Duchaine muscular dystrophy. It's both the most common and most severe type. It's carried by females and expressed in males. It's usually diagnosed anywhere between the ages of 2 and 10. And death usually occurs from respiratory failure sometime in their 20s. Now, there's no current cure for this, but there are some therapies being experimented with. Okay, last slide here. The female skeletal muscle makes up about 36% of the body mass, whereas in males it's about 42% of the body mass. Now, what you have to understand is that if we took a square unit, right, of skeletal muscle in a female versus a male, the strength is the same. It's not that male skeletal muscle is stronger. It's that males have a higher percentage of skeletal muscle, and that is due to testosterone. So males have about 20 times the testosterone of females, and so they're going to produce a larger percentage of that body mass being skeletal muscle.